Dear students, in this session we shall discuss about ion selective electrodes. Under ion selective electrodes, what are ion selective electrodes, application of these electrode and construction and working of glass pH electrode, and using this electrode how the pH of a given solution is determined will be studied. So, observe that ion selective electrode, the name itself representing these electrodes select a particular ion in a mixture of ions and develops a potential, then that type of electrodes are known as a ion selective electrode. That means ion selective electrodes can interact more selectively with a particular ion in the mixture of ions while neglecting the ions. This type of electrodes are known as a ion selective electrode. So, therefore, these electrodes are used to measure the concentration of particular cation or anion in the mixture. These electrodes can measure very dilute solution like 10 to the power minus 6 molar solution can be measured accurately using these electrodes. So, therefore, these electrodes are more selective and they are suitable for the rapid, low cost and accurate analysis. So, that is why these electrodes are used in almost all the fields like agricultural, biological, chemical, clinical and industrial laboratories. So, best example for ion selective electrode is a glass pH electrode. So, observe that the best example for ion selective electrode is a glass pH electrode. So, this glass pH electrode is more selective towards H plus ions or pH. So, therefore, these electrodes are used to measure the pH of a given solution. That is why these electrodes are known as a glass pH electrode. That means these electrodes are more selective towards H plus ions or pH in the solution containing mixture of ions. Hence, it is used in the measurement of concentration of H plus ions and pH in the solution. Electrode, this diagram represents the representation of a glass electrode. Observe this glass electrode. This glass electrode consists of a long glass tube having small glass membrane bulb at its bottom. Then silver chloride electrode is inserted in this electrode. It acts as an internal reference electrode. The role of this silver chloride electrode is it is used to measure the membrane potential as well as it is used to connect this electrode or couple this electrode with other electrode. That means it is used for a electrical contact. Observe that this glass bulb is filled with 0.1 molar HCl solution or a known buffer solution. Glass pH electrode, it consists of a long glass tube having small glass membrane bulb at the bottom, and silver chloride electrode is inserted in this. This electrode acts as a internal reference electrode and it is used to measure the boundary potential, and it is also used to connect this electrode with other electrodes. Let us discuss the working of this glass electrode. So, generally in this type of electrode, it is having a glass membrane bulb. Through this glass membrane bulb, there will be an exchange of ions. That means, when this glass electrode is placed in a solution of unknown concentration, then through this membrane, there will be exchange of H plus ions. Due to this exchange of ions at the interface of the membrane, it develops some potential. That potential is known as a boundary potential or a membrane potential, and it is abbreviated by the letter EB. This potential is measured by using a silver chloride electrode. So, observe that the concentration of inside solution. So, that is considered as a H plus ion solution of 0.1 molar concentration. Let us consider this one as a C1 concentration, and this one is dipped in a H plus solution of unknown concentration. Let us consider that concentration is C2. Observe the working of this electrode. So, this inside also contains H plus ions, outside also H plus ions are present. So, due to difference in concentration gradient, there will be a exchange of ions inside and outside through this membrane. So, due to this exchange of ions, it develops a potential at the interface of the membrane. That potential is known as a boundary potential. So then what is the expression for boundary potential? So boundary potential is abbreviated by EB. So it is nothing but, so observe that the solution inside and outside is same, only the concentration of this H plus ion is different. So therefore it is nothing but it can express, it is working as a concentration cell. What is the expression for concentration cell EMF? EMF or a potential of a concentration cell is measured by using the formula. Recall the formula, 0.0591 divided by N log base 10 of concentration of C2 divided by concentration of C1. So observe that, so during the exchange of this ions, it acts like a concentration cell. Concentration potential is given by EB is equal to 0.0591 divided by N log base 10 of C2 divided by C1. What is the value of N for H plus ions? The number of electron involved in 1. So therefore this expression is reduced to 0.0591 log base 10 of C2 minus 0.0591 log base 10 of C1. C1 represent the concentration of H plus ions inside the membrane. Its concentration is always remains constant. Its concentration is 0.1 molar. So therefore I am considering this whole term as a L. So therefore EB is equal to L plus 0.0591 log base 10 of concentration of C2. So what is the L value? L value, observe that L is equal to minus 0.0591 log base 10 of C1. Why it becomes constant? Because always the concentration of H plus ion inside the membrane remains constant. So therefore this whole term is considered as L. So therefore this equation is reduced to EB is equal to L plus 0.0591 log base 10 of concentration of C2. What is C2? C2 is the H plus solution, that is hydrogen ion solution of unknown concentration. So I am altering this equation like this, L minus 0.0591 minus log base 10 of C2 is nothing but H plus ion concentration. Observe that I am altering this equation. So minus into minus become plus only. So therefore L minus 0.0591 minus of log base 10 of H plus ion. So minus log base 10 of H plus ion is nothing but it is pH of the solution. So therefore this equation is changes to L minus 0.0591 pH. So this is the expression for boundary potential. What is the boundary potential expression? EB is equal to L minus 0.0591 pH. What is the definition for pH? pH is nothing but minus log base 10 of H plus ion. So therefore instead of this, so here we written it as pH. So therefore EB is equal to L minus 0.0591 pH. So this gives the expression for boundary potential. EB is equal to L minus 0.0591 pH. Is it clear? So what is the potential for glass membrane? So glass electrode or a glass membrane boundary potential or a membrane potential is nothing but it is L minus 0.0591 pH. When glass electrode is dipped in a solution of unknown solution, it develops boundary potential. So observe that when the concentration of this, what is the expression for boundary potential? EB is equal to 0.0591 divided by N log base 10 of C2 divided by C1. So observe that when the concentration of C1 is equal to C2. So what is the answer for EB? C1 is equal to C2 means log 10 value is 1. So log 1 is, log 1 value is 0. So, therefore, EB value is 0. That means theoretically when the concentration of H plus ions inside and outside is equal, then according to theoretical EB value should 0, but practically EB is not equal to 0. That means it develops a small potential that potential is considered as a asymmetry potential and it is abbreviated by ASY. What is asymmetry potential? It is a small potential developed when the concentration of C1 is equal to concentration of C2. Why? It develops a small potential because observe this, the radius of curvature of this glass bulb is different inside as well as outside. So, therefore, H plus ion activity is different. Due to this difference in H plus ion activity, it develops a small potential. That potential is considered as a asymmetry potential and it is abbreviated by the letter ESY. So, this value depends upon time to time and its value is also depends upon electrode to electrode. Next, observe that this glass electrode consists of a reference electrode. That reference electrode, internal reference electrode is a silver chloride electrode. So, therefore, it also develops some potential. That potential is considered as a E reference potential. So, how the glass electrode is represented? First, we have to represent the internal reference electrode. Here the internal reference is silver chloride electrode. So, it is constructed like this. A platinum wire is first coated with silver and then with silver chloride, then it is in contact with 0.1 molar HCl solution. So, therefore, representation is Ag. What is the physical state? Solid, which is in contact with AgCl. So, physical state is also solid. Then it is in HCl solution. What is its concentration? 0.1 molar. Then observe that through this glass membrane, so there is an exchange of ions. So, therefore, this is the glass membrane or simply glass. This is the general representation of glass electrode. So, therefore, due to this, the potential developed at the glass electrode is nothing but it is the sum of glass electrode potential is abbreviated by G. It is the sum of electrode potential developed at the boundary, that is boundary potential, plus reference electrode potential, that is EF, and a small potential developed when concentration C1 is equal to C2. So, it is asymmetry potential. So, therefore, the potential developed at the glass electrode is the sum of boundary potential, reference potential and a asymmetry potential. Substitute the value of boundary potential. So, what is the expression for boundary potential according to this expression? It is L minus 0.0591 pH plus E reference value plus asymmetric potential. Observe that L remains constant, reference electrode potential value remains constant and asymmetry potential also remains constant for a particular electrode. So, therefore, these three constant becomes another constant. Let us consider that constant as E naught G minus 0.0591 pH. So, what is the expression for electrode potential of glass? Electrode potential of glass is nothing but it is E naught G minus 0.0591 pH. By using this equation, we can easily calculate the pH of any solution. So, observe that E naught G is a combination of three constant. What are those constant? L plus E reference plus E asymmetry, where E naught G is a constant. It is a combination of L plus electrode potential of reference plus asymmetry potential. This is calculated by placing this glass electrode in a solution of known buffer. Then we can easily calculate the value of E naught G. Then next we will discuss about the determination of pH of a given solution by using glass electrode. Next one is measurement of pH of unknown solution using glass pH electrode. So, to measure the pH of a given solution or an unknown solution, first we have to construct the galvanic cell. How to construct the galvanic cell? Glass electrode should coupled with a saturated calomel electrode. Then these two are connected by a highly sensitive voltmeter. So by joining these two cells by voltmeter, we get a galvanic cell. So then how this galvanic cell is represented? Observe the diagram. According to this diagram, the glass electrode is coupled with a saturated calomel electrode. Then this galvanic cell assembly is dipped in a solution of unknown pH, or it is also known as a test solution. We have to find out the pH of this test solution by using this galvanic cell. Then how to represent this galvanic cell? According to the diagram, so calomel electrode acts as an anode and the glass electrode acts as a cathode. So therefore first we have to write the calomel electrode representation. How the calomel electrode is represented? So mercury, which is in contact with mercurous chloride formula is Hg2Cl2, then it is in contact with saturated KCl. So observe that this porous plug acts as a salt bridge. So therefore salt bridge is represented by two vertical lines. So this represents the representation of anode. So according to that diagram, so saturated calomel electrode acts as a anode. So anode representation is Hg. So its physical state is liquid and mercurous chloride is a solid. Then KCl is saturated KCl. Then next represent the representation of a glass electrode. So observe that the H plus ions are exchanged through this unknown solution of unknown solution. So therefore we have to mention here as test solution. So through the glass membrane there will be a exchange of H plus ions through this test solution and the glass. Then we have to represent the reference, internal reference electrode is a silver chloride electrode, that is HCl 0.1 molar, then silver chloride, then silver. So physical state, this is a solid, this is also solid. This is the representation of this galvanic cell. According to this diagram, so calomel electrode acts as an anode. So therefore first we have to mention the anode representation. Then glass electrode acts as a cathode. So therefore we have to represent the cathode. So cathode is the glass electrode. Its representation is glass, HCl, AgCl and Ag. So exchange of ions takes place this test solution. So this is unknown pH. So how the EMF of this galvanic cell is calculated? It is calculated from the equation, that is E cell is equal to electrode potential of cathode minus electrode potential of anode. So according to the diagram, the cathode is a glass electrode. So therefore Eg minus E saturated calomel electrode. Saturated calomel electrode is represented by SCE. SCE means saturated calomel electrode. So observe this expression. By knowing the concentration, by knowing the potential of saturated calomel electrode and E cell value is obtained from the voltmeter, then we can easily calculate the electrode potential of glass. We already known that electrode potential of glass is nothing but it is the sum of potential of boundary potential and reference potential and the asymmetry potential, and simplifying this we get the expression Eg as Eg naught minus 0.0591 pH. Substitute that value here. What is the electrode potential of glass expression? It is E naught G minus 0.0591 pH minus electrode potential of saturated calomel electrode. So what is the aim? So we have to find out the pH of unknown solution. So therefore express this equation in terms of pH. So therefore this equation becomes 0.0591 pH is equal to E naught G minus E saturated calomel electrode minus E cell value. So, express this in terms of pH. So, therefore, this equation is reduced to E naught G minus E saturated calomel electrode minus E cell divided by 0.0591. This is the expression to calculate the pH of any given solution or an unknown solution by using this expression, that is pH is equal to E naught G minus saturated calomel electrode potential minus E cell value. E cell value is got from voltmeter, then E saturated calomel electrode is known to us, its value is 0.2422. Then E naught G value is calculated by placing this galvanic cell assembly in a buffer solution of known pH. So, by substituting all these value and it should be divided by 0.0591, then we can easily calculate the pH of a given unknown solution. So, therefore,